What we're looking at in this presentation is the concept of the dihybrid cross as opposed to the monohybrid cross, which looks at one trait, whether it's color or height or eye color or something like that. In the dihybrid cross, we're actually looking at two traits at the same time. You might need up to 16 squares in your Punnett square in order to solve this. You might and you might not. So what you're looking at here is a dihybrid cross in which they're looking at P's and they're looking at two traits. So they're looking at texture, whether it's round or whether it's wrinkled. And they're looking at color, so whether it's yellow or whether it's green. So it's taking two parents, figuring out what the gametes are, and then doing the cross and seeing what the probability for the offspring are. Okay, so I want to look at the dihybrid cross now, as opposed to the monohybrid cross, which is looking at one trait. The dihybrid cross is looking at two traits. I have made a 16 square Punnett square. Depending on the question, you may not need this many squares, but for the first problem I'm going to do, you do. So the first thing we'll do is we'll look at a cross between two organisms that are both heterozygous for both of their traits. So I want to look at two traits. I'm going to look at height and I'm going to look at color. So for height, I'm going to use a big T to mean tall because it's a dominant trait. And I'm going to use a small t to be for short. These are pea plants, by the way. And for color, I'm going to use a big Y for yellow and a small y for green. So what I want to do is I want to cross two pea plants that are both heterozygous for both traits. So I'm going to have a tall, heterozygous, yellow pea plant, and I'm going to cross it with one that is exactly the same. That's usually step one when you're doing a dihybrid cross, is find the information in the question and just write your cross out. It helps you get organized. So the first thing I'm going to do here now is meiosis. So meiosis breaks apart these paired traits, and you only have half in the gametes, so that's what I'm going to do here. I'm going to try to find the gametes for one parent, put them on the top, and the gametes for the other parent, and put them on the other side. So I'm going to start by just looking at all the different ways these can sort out. So when meiosis happens, the gamete's going to get either this T or this T, not both, and it's going to get either this Y, not both. So this T could sort out with the big Y. So big T with big Y is one of my possibilities. Now this big T could also sort out with a small y. The other thing that could happen is I could get this small t sorting out with a big Y. It's also possible that I could get this small t and a small y. Now I really don't have to do anything for this one because both parents are exactly the same, but I will. I'm going to do it again just so you can see, make sure you understand what I did. So big T with big Y, write it down. Big T with little y,
Oops, I should have wrote that one here. And then I could go little t with big Y. And little t with little y. Note the gametes for both parents are exactly the same because they started out exactly the same. That isn't always the case. So what I want to know essentially is what happens when I cross a tall yellow heterozygous plant with another tall yellow heterozygous plant. What possibilities are there for offspring? I'm going to do a couple with you, and then I'm going to shut it off, and hopefully you'll sort it through yourself and then pick up again. Okay, so let's just start here. This one is going to get a big T from this parent and a big T from this parent. Going to get a big Y from this parent and a big Y from that parent. So conventionally, you would usually go in alphabetical order. So put your T's first in this case. And you always keep like alleles together. Like it wouldn't be wrong, but it's just not the way it's commonly done to go TY, TY. Just keep the homologous chromosomes together, I guess. So let's just pick any other random one. I'll pick this one here. So this one, it's got a T, a little T from that parent and a little T from that parent. And it's got a big Y from that parent and a big Y from that parent. Okay, so now your job is to go ahead and to fill in the chart. I'm going to stop my video, fill in the chart, and then we'll pick it up again. Okay, so I filled in my chart. Hopefully you got yours filled in the same. So what I want to do now is I've written up the forms of the traits here just so we can remember what we're dealing with. And I also wrote down all the different combinations that the offspring could be. They could be yellow and tall, getting both dominant traits. They could get the dominant yellow with the recessive short. They could get the recessive green with the dominant tall. And they could get both recessive traits. So what we want to do now is just go through our chart and count up how many of each there are. So when I'm looking for yellow and tall, I'm looking for everything that has at least one dominant yellow and at least one tall. And so I'm going to go ahead and circle those. So let's start here. Well, this one, yes, this would be tall and yellow. This one has at least one Y, so it's going to be tall and yellow. This one looks tall and yellow. Yeah, this one has at least one of each, so it's tall and yellow. Again, tall and yellow. Tall but not yellow, so I'll skip that one. Yeah, this one's tall and yellow. Tall but not yellow. Tall and yellow. Tall and yellow. Yellow but not tall. Yellow but not tall. Tall and yellow. Tall but not yellow. Yellow but not tall. Neither. Okay, so I have 9. 9 out of 16 are yellow and tall. Now let's go ahead and figure out how many are yellow and short. So to be short, they're going to have the 2 recessive T's, and they're going to have at least one big Y. So recessive T's, yes. Big Y, yes. One there, and one there. I don't see any other ones. These ones are going to be short and yellow. Okay, so that's 3 out of 16. Okay, now we want to look for the ones that are going to be green and tall. So they're going to have at least one big T, but no big Ys. So here's one, has at least one big T and no big Ys, okay. This one, and also this one. So we've got 3 out of 16, and then we have one left, and it has both the recessive conditions. It is short and green, so that's 1 out of 16. So we have a 9 to 3 to 3 to 1 ratio. And that's a constant. It's always going to be 9, 3, 3, 1 anytime you cross two organisms that are heterozygous for both traits. So you'll probably get that question at one point, and now you know how to do it. Sometimes you'll get a question with differing alleles, and you won't find that you need all of the 16 panes. We'll try another one like that next. Okay, here's a problem. Cross a homozygous tall blue-eyed man with a short brown-eyed woman who is heterozygous for her eye color. Remember I said the first thing you're going to want to do is set up your cross so you know what you're dealing with. So, this man is heterozygous tall. And I'll just insert a little footnote here that human height is not a Mendelian trait like tall or short, but I'm just making that up. Okay, so we'll say tall man with blue eyes, so we know that's his genotype right there. He's tall, homozygous, tall, and he has blue eyes. And we're going to cross him with a woman who is heterozygous for eye color. So she's got brown eyes. Oh, she's a short woman, first of all. So she's got 2 little t's, and she's got a big B and a little b. Okay, so there's our cross right there. And that would be the very first thing I would do, is write down the cross.
Oh, yes.
Okay, I've set up my 16 square Punnett square, but I'm thinking I don't need to use all 16 squares. So let's start with the first parent here. This was Dad, who was tall and had blue eyes. Okay, so how are all the different ways these alleles can sort out? Well, you can have big T with little b, or big T with little b, or big T with little b, or big T with little b. So you see, there really is only one combination of alleles that he can produce, and he's going to have big T with little b. So there really is no point in filling this in the rest of the way. We just simply don't need to. It's just redundant information. So, because they're all going to be the same. So now for Mom, we're going to have little t with big B, okay. Little t with little b. And then the same thing all over again, little t with big B, little t with little b. So there really is no point in doing that again. It's going to be the exact same information. So I don't need more than 2 Punnett squares to do this problem. So what are the possibilities for the offspring? I've got big T with little t, big B with little b, or big T with little t and 2 little b's. So what are the possibilities for their offspring? Well, their offspring are going to be tall for sure. See, this possibility is tall, tall. They're carrying a recessive allele for shortness, but they won't be short. But their offspring could be brown-eyed with heterozygous brown eyes carrying the blue, or they could be blue-eyed. So their offspring can be tall and brown-eyed, or tall and blue-eyed. And those are the only two possibilities, and it's a 50-50 chance either way.